I don't know if anybody has. I don't know if anybody has any announcements that they would like to make before I introduce Steve. Feels so wrong to just start Herp group just like this. You know, it's so different than it's been traditionally. But
there was,
your take.
Jim, there was some support from people for like an abbreviated journal session. So if you have anything you want to mention, I think everyone's on board.
I don't have anything that I want to mention. I came unprepared.
I do have one, which actually.
Okay, let's do it. Excellent. Back to literature.
I think we're going to cover it on AmphibiaWeb, but it was this paper about the small genome in frogs. It was called a bird-sized genome, and we all decided it's named that way because Scott Edwards is an author.
An ornithologist's point of view about that tiny genome.
Exactly. I mean, it's the large genomes of amphibians have been a problem, as everyone else has experienced the genome revolution. Us amphibian people are kind of a little left behind. I'm going to post the link in the chat because I can't claim to have read the whole paper.
Yeah, the genome size was around 3 gigs.
Hold on. I don't even remember the number. Must be less than that.
Must be less than that because that's not really bird sized.
It's human.
Yeah.
1 gigabase.
Oh, it's a 1 gigabase genome.
Yeah, I just put it in. So Awake was the handling editor for this.
Right.
So look out for we'll have a news box from AmphibiaWeb on this at some point.
Cool. Does anybody else have any impromptu literature they'd like to present? Trying to like think about what I've seen lately. Nope, nothing available. Next time. Now that I know that that I'm allowed, I'll bring literature next time. Does anybody have any announcements that they'd like to make to the Herp group crowd? No. All right. Well, I think that we can then go to the main event, which is tonight's speaker, Steve Poe. Known Steve for a long time. We were grad students together at the University of Texas, and Rafe, of course, was also there, and Catfish was there too, I think. He was our advisor. Yeah, Chris Austin was there. My wife Sharon was there. You know, our little cohort is well represented here at Herp Group. Sharon's in the background here as well. Steve did his undergrad at Harvard, and Ernest Williams was his, original mentor. And so Catfish kindly showed us a picture of Ernest there, one of the fathers of Enolas taxonomy and one of the most famous practitioners of anole biology. And after Steve graduated from Harvard, he joined the Canatella and Hillis labs at, you were also Hillis sponsor, weren't you, Steve?
I think his name was in there, yeah.
But really, we were in the Canatella lab, and where he did his PhD work, on anoles, and then after Steve finished off his PhD, he actually, before he finished his PhD, he got his faculty position at New Mexico, which is pretty awesome. And he also had a Miller postdoctoral fellowship here at UC Berkeley, and so he came to Berkeley and was co-sponsored by Marvelly and Dave Wake. But he already had a job waiting for him at New Mexico. So after his Miller was completed, then he was off to his current position. Steve is Steve and Jonathan Losos must be the two people in the world that know the most about anoles. I'd be willing to venture that.
I don't know.
Steve knows a lot about anoles, and so and he's published tons of papers about them and various other things as well, a lot of theoretical papers as well. But I asked him specifically if he could tell us about his field work. I periodically check out his website. He doesn't know this. Just to before he used to have a lot of pictures of his field trips. Now he just has kind of like one shot for each field trip and maybe a little caption. But there were some additional photos there in the past. So I was really hoping that I could convince him to give a talk about his work because he's done field work all over tropical America studying anoles. And I think that's going to be the main event tonight. And Steve, thanks so much for being willing to give this Herp group. And the floor is yours.
Oh, thank you, Jim. Yeah, it's great to be here, even virtually. Like, it's just so amazing to see the folks here. it's just, I mean, I had such a great time there, you know, a long time ago at the MBZ, in Marvelie's lab and in Dave's. And hey, Dave, it's great to see you. And
Wow.
it was just, you know, it was just about the best academic experience I've had as the couple of years that I was there. That, you know, they have such incredible labs and incredible people in them, and so just, you know, learning from the people there at MBZ and being immersed in the herp crowd there was just incredible for me. So it's great to be back, even virtually. So let me see if I can. So I share the screen now. Is that correct?
That is correct. As
Okay.
long as you've been authorized to do it, which I think Becca can authorize you if you aren't already authorized.
Okay. Is that let's see. Did that work?
Yes,
Can you see my, you can see my slide?
totally.
Okay. Cool. All right. Before we get started, two things. One, I'm out on my porch here in Albuquerque. So if you hear a siren, don't worry about it. It's probably just some really serious crime being committed close by. And the other thing is that you're making me talk at 7:30, so you're going to get the Cabernet version. All right. All right. Let's start with a quote. Anybody can catch a lizard, and it's catching that particular lizard you want that may be difficult. High up on a sodden, windy peak with a thunderstorm visibly and rapidly approaching to windward, a magnificent specimen of some montane rainforest form swirling farther and farther out in the cackling trunks over a great abyss can lead to intemperance in a man. So this is a quote from the great Skip Lizzell, a fantastic field biologist who was associated with the MCZ and then ran a conservation firm for a long time. And this is from his 1972 monograph on the Lesser Antillean anoles. So he's making this comment actually about catching anoles. And that's what we're going to be talking about today. We're going to talk about the techniques involved. We're going to talk about some of the places that we've been with my lab, talk a lot about just cool anoles, and really you'll see there'll be a focus on those species that have caused a great deal of intemperance in my lab. All right, but let's get some context first. So everyone here knows that herping is a real thing, and it means looking for reptiles and amphibians. And like any other kind of animal search and capture approach, it's the field has developed its own specialized set of techniques. So one of the most venerable of these, of course, is road cruising. You know, driving roads, paved roads, usually at night, looking for snakes that have come out on the road nocturnally to forage or to move around, or to see snakes that, you know, are on the road after they've just been out diurnally. And so this is an approach that I don't know if it was pioneered by this person. Many people at this talk probably know who this mad scientist is. Of course, that's Lawrence Klauber. And he wrote this fantastic paper, 1937, about night collecting in the desert with ecological statistics. And it really gives a sort of how-to guide, a primer for how to road cruise for snakes. And it's just amazing. And it includes, you know, a bunch of data. There's tables in there. And it's just an incredibly useful piece of work. I mean, you can look at his table and be like, okay, you know, if Lawrence Klauber never found a coach whip driving at night, probably I'm not going to. You know, that's probably something I should be looking for during the day. And so this piece was a big inspiration for a chapter I've written about finding anoles. And you don't have to read this at all. It's on my website. You don't have to read it because I'm going to give you all the information from it today. Okay, another specialized herping technique, of course, is flipping debris. You know, herpetologists love to find trash heaps, you know, sheets of corrugated tin, boards. We like to go and try and flip these over and look for snakes a lot, but also really just about any kind of herp can be found underneath things. Noosing is another long-standing approach, which involves having a rod and then a loop on the end with a small loop that you put a bigger loop through so it slips, and you put it over the lizard and jerk back and catch them by the neck. I've seen in review a couple times that this should now be called lassoing, which probably is a great idea. And then for tropical herpetologists are really familiar with the idea of spotlighting. So going out at night and having a bright light. And then amphibian biologists especially will use this technique to find things out active, but then also, you see a lot of things sleeping as well. So today there'll be a big focus on spotlighting. Okay, in a talk about looking for a very particular kind of anole, why do I have a picture of disgraced cyclist Lance Armstrong in my talk? What I'd like to do is draw an analogy here, which may or may not work. What Armstrong is really known for, first of all, is doping, right? So you probably think I'm going to talk about like taking drugs to stay up all night or something. That's not where this is going. The thing that Armstrong is known for just as much within the cycling community is his specialization. So, pre-1980, this really started with Greg LeMond, I'd say, who realized that the United States only cares about one bike race, and that's the Tour de France. And so these are two Americans, LeMond and Armstrong. Before that, the great cyclists, Eddie Merckx and Bernard Hinault and Coppi and all these, they're trying to win every race all year long. And Armstrong, his entire season, everything he did, all his training and all his racing was geared towards winning that one race, the Tour de France. So the analogy I'm drawing here is that we take all those techniques for herping and we strip them all down to just the anole parts. So everything we do on our field trips is about finding anoles. We might stumble across frogs, we might see a snake once in a while, but we're doing everything we can to find anoles in particular. And our goal is this, we're evolutionary biologists, we're taxonomists. So we generally are trying to efficiently collect as much Anolis lizard diversity as possible. And here I'm mainly talking about species diversity, but also, all kinds of biodiversity that you can think of. So we operate under two principles here. One of these is targeting. As I've already kind of mentioned, we focus our collecting on anoles, and in particular, we focus our collecting effort on rare anoles. So when we went to Ecuador the first time, first thing we did was go down the slope of the Andes to try and find Enolas orsesi, which was at that time known from only 2 specimens. And the idea is that if we focus on these rare things that are expected to be difficult, well, we should be able to get the commoner things as bycatch as we go along. And then the specialization part. Here's where we herp almost exclusively at night. And I'll give you more information about that as we go forward. But we'll go trips where we'll go the entire trip and we won't catch a single lizard during the day. And then we also use one particular technique, and that's spotlighting, with also what's colloquially has come to be called, one of my students came up with the name a shake and bake, and that refers to knocking a lizard down from a tree and catching it. And so we'll go an entire trip and we won't noose a single lizard because this is developed for us to be the most efficient way to do things. So why herp for anoles at night? There's 2 big reasons. And one of them has to do with the biology of anoles and really how they sleep. So this is a forest in Belize. This is in the Maya Mountains. It's actually around some Mayan ruins. And you can see, what do you see here? You see a dark scene, and then this lizard just screaming at you because you can see the contrast there of this Enolas lemurianus jumping out. And you can also see, this thing is 1.5 meters off the ground. You can walk up and grab it. You don't have to be an expert with a noose. You can just go get it. And anoles sleep, almost all species of anoles. I'll talk about some exceptions later, but almost all species of anoles sleep on really light vegetation, twigs or leaves. You'll never find one sleeping on vertically on a big tree trunk. And the reason they do this is, it's thought that, okay, if their perch is jiggled, they're main nocturnal predators or snakes. The snake crawls up that the root or the shoot of that palm and jiggles the leaf, and the lizard detects it and drops it and can get away. And so it's supposed to be this predator avoidance thing. The other reason that we go at night is this, and the reason is that if you want to catch common anoles, you can go during the day, you can go during the night. If you're in Florida and you want to get Segra, you want to get Carolensis, it doesn't matter. Knock yourself out however you want. But if you want to catch rare anoles, you really have to go out at night. And this was encapsulated nicely by the great naturalist Ken Miatas in his description of Enolas lynchi. He says, until recently, there were very few representative series of most South American anoles. Moderate to large series of some species from the Andes and their western and eastern flanks have been collected during the past decades by workers interested primarily in frogs, who find sleeping anoles at night. They have sometimes been the ones most poorly represented in museum collections. In some cases, it seems as if a different anole fauna can be found at night. And so that different fauna is the rare things, it's the difficult things. And so some of the kind of people who made this possible are some of the amphibian biologists shown in the lower left there, John Lynch and Linda Trube and Bill Duhlman. And that's some of the people Miata was really talking about. And on the right, maybe lesser known, Richard Thomas is at the University of Puerto Rico. Now, just to give you a kind of vignette that tells you about him and also tells you about this whole approach, these Enola species, Enolas occultus, was described in 1965 by Juan Rivero and Ernest Williams. And the first specimen of that species was found in 1963 by Rivero's wife and child. They just stumbled across it. And keep in mind, Puerto Rico was Puerto Rico. It had been herped to holy hell by the time the 60s rolled around, right? So to find a new anole at that point was crazy. Nobody found another occultus for another 2 years. Finally, one more was found. Then Richard Thomas got to work and found the next 34 specimens within less than a year. How did he find them? He went out at night and looked on dead vines and twigs. So if you look just to the side of Thomas there, that's where occultus always sleeps, is on a twig like that. And so you can see where, that's a lizard that's going to be hard to see. And even at night, it's not that easy unless you know kind of exactly where to look and how, like Richard Thomas did. Okay, and so I don't want to give the impression that we're breaking ground here or reinventing the wheel or doing something that every other tropical biologist doesn't know about. This is an article from 1923, Popular Science. It's actually writing about a trip by Noble, G.K. Noble, the American Museum curator. And so flashlights reveal frog monsters, and they've got all this, they're finding all these crazy things because they're out at night. How did I lose? What happened there? I think there was an insect. Okay. All right. So just to make the point that I'm talking about stripping things down, but we're not really doing anything original here. We're just taking things that are always done and doing them in a way that best fits our needs. So if you go on a kind of herp trip where you're doing something like a general survey, like a BioBlitz or something like a herp class, you're probably going to get up in the morning, try and get some lizards. You're going to go look for trash heaps and flip stuff in the afternoon, maybe go cruise for snakes at night, and then get to bed in the early morning. On our trips, we generally get started working at about 6 in the evening, 7 when the sun's going down. And then we almost always work all night, so work until the sun comes up. The idea being that if you have this kind of carrot in front of you to finish the night, you're much less likely to kind of reason yourself out of looking. We've all had those nights where it's like, oh, they're just not out tonight. It's who knows what's happening. And so you stop, and then if we kind of like make it this thing where we have to be up the whole time, then then sometimes you find something really cool at 5 or 6 in the morning, and then we sleep from about 7 or 8 in the morning until the afternoon. And so of course doing this results in this isn't like a natural schedule for humans, right? And so people fall asleep in the car all the time, like Kevin McInnes in the upper left. People like Eric Schad on the middle left fall asleep in the hotel while charging their batteries. Eric Hulebeck on the lower left fell asleep on the trail on the way to the Darien. And some of us are even able to sleep in a Pecky boat during a 7-hour ride up a tributary of the Amazon. That's my buddy Christian Yanez Miranda with me in the boat there. But of course, this also opens you up to all kinds of hijinks. Like upper right is my TA Julian Davis passed out on the bus stop in Boquete. So of course the students thought it would be hilarious to put an Enolas microtus on her face, and she didn't wake up. This is me passed out on the drive from Cali to Medellin. And while I'm passed out in the middle seat of the van, apparently I decided to drink a beer. And then also for some reason decided to smoke a cigarette. But that couldn't even be my hand, right? So all right, so let's talk about the work a little bit, and leading into that, talk about some of the equipment. So this is Ian Latella, who got a master's in my lab, and he has our basic equipment with him. So he's got, you can sort of see in his left hand, he's got a pole, and that's a literal 20-foot pole. That's a telescoping panfish rod that can go out. So it's kind of gotten to be our go-to pole. We've tried a lot of different things, you know, golf ball retrievers and stuff like that. But this pole is the one that we keep going back to because it's pretty cheap and it works really well. And yeah, we'll bring, you know, 4 or 5 of them along with us. And then for lights, of course are really important. What we've kind of settled on are these actually night mountain biking lights. So this is a Dyno XML3 here. I think it's a 2000 lumen. And the combination of, it's very light, and it's, you know, I don't like headlamps because you have to be able to move the light around and see the contrast in different ways. You know, you can put this in your mouth when you want to grab something. And the kind of the throw, which is how far the light goes versus the flood, is a really nice ratio for a lot of these if you get the right model. Now, this isn't to say that we don't like go supercharged in some cases. So this is an Ace Beam K30 GT. You know you have a serious light when it's got a rifle mount. And this is actually a 5500 lumen night, but of course the key is not the lumens, it's the candelas, right? So this is 265,000 candelas, a throw of over 1,000 meters. And this one,
can you say the name of that one again? Sorry, I just missed the name.
Yeah, it's an Ace Beam K30 GT. And so the downside to this, you know, is that it's heavy and it's hard to, you know, when I use it, I put something over this so I can put it in my mouth, but it's still really heavy. But it's in terms of the throw versus the flood, this has just about the perfect fit for it. You know, if you have too much throw and it's too narrow, then it's too hard to search for things. If you have too much flood, then, you know, you're just washing it out like daytime, which isn't good either. And those don't, if there's a lot of flood, it doesn't throw as far. So this is like, if I were to kind of optimize the light, this would be it, but the other downsides of it keep me from using that as my main light. So we think about lights a lot, as you can tell. I noticed these, I had special glasses made. I had eye surgery a few years ago, but I learned that I could actually get my eyes a little bit better by getting these high-end glasses that improve by like 1/4 of a diopter with a slight orange tint so you can see contrast better. But all right, let's try this out. Can anybody find the lizard in this scene? Jimmy? Okay, so this is Enolas biporcatus. Got away in the hotel along the canal there in Panama. You know, this is, I'll show you the 2, you know, places that you see anoles out sleeping most commonly. These are leaves. A lot of things are fern specialists. So this is Enolas etheridgei in the Dominican Republic. The other main place is twigs. So you can probably see this Enolas polylepis, you know, in the middle right there. But of course, you know, a more standard scene is something like this. This is a roadside in Panama, north of Boquete. And this is the pretty common, let me not really digress, but tell you a bit about the kinds of habitat we look for. You know, so pristine habitat is great, and you have to herp it when it's available, right? Because everything evolved in pristine habitat. But, you know, if you go looking for anoles in Amistad National Park, walking their trails, it's going to be really hard to find them. And I've had multiple cases where we go through the great habitat, we always try and herp it, and just do terribly. And then we come out and we hurt the roadside next to the good habitat, and then that's when we just kill it. So we tend to like good habitat that comes up to crappy habitat. And that seems to be the best place. And along with that, just as an aside, I don't know a single anole that requires pristine habitat to live. If the trees are there, they'll be there. So this guy, so this one's right there. If we pan out, you can see both the contrast, the body. This is Enolas kemptoni, and you can also see the tail coming down, right? So this is kind of, you know, what we're up against and what we're looking for. Okay, so we found the lizard. What do we do now? Let me set up this next scene. So this next scene is, it's in Puerto Rico. We're at a bridge, and we're going to do a shake and bake here. So my colleague Tom Kennedy, who just as an aside, all the good anole photographs in here are by Tom Kennedy. He's a faculty member here, and he's a tremendous photographer. But he's come with me on a bunch of trips. So we're at this bridge in Puerto Rico. If the sound works, you'll hear the coquis. I'm gonna lift a pole up, smack the lizard. If you look closely, you can see the lizard fall behind me, and then you'll see Tom catch it. All right, so let's see if this works.
Editing is for
Okay, so that's me. Got the pole. You hear the cokies. And Matt, there it comes. And what's the next step? So this is one like this one I debated whether to include this with this crowd. This isn't a crowd here that needs to be convinced of the value of specimens and why we do this sort of thing. But I have talked about this stuff before, and this is the part where people always get this, wait, what? And hold on a minute. And this is a very understandable view from the public, right? That's you want to save this species, yet you're killing them. And so a lot of kind of public versus biologist conflict has to do with that level of biologists generally care about the populations and the species. The public generally cares about the individual. And so it's a legitimate conflict that we have to be really aware of philosophically, and we have to care about and take seriously. But that view then has even, this paper was when it kind of hit us that it seeped into, some environmental philosophers and even some biologists. And so this, of course, again, everyone here probably is familiar with both this paper and the reply to it, which was kind of trying to lay out a bunch of reasons that I won't, again, bother with this group with, but, why it's important to preserve specimens and keep them. And I would say this is probably the best paper that the three of us, Jimmy and I and E.O. Wilson, ever wrote together. Wouldn't you say, Jimmy? I think this is probably our best. So but the thing about this is the theory here is so clear to us as biologists, right? The demography, the argument that, okay, they're little things and there's a lot of them. So whether you take 5 or 10 or, get all Doolman on it and take 1,000, it doesn't matter, and we know that based on, what we know about these populations. But that's actually a really hard thing for, again, people who don't do what we do, including a lot of permit granting societies. Like, how many of you have had the experience where you say, okay, I would like to take 15, and you get the response back saying, why don't you take 5? And you're thinking to yourself, 15, 5, come on. And so what we thought is, we should do something practical that actually tests this kind of, is there an effect to herpetological collecting? And we do this there's a lot of neat data we get out of this, so it's not just for this, but we wanted to do an experiment that showed that if you go through and just collect the heck out of an area, we wanted to see how long it took that area to bounce back. Would it be in a week? Would it be in, 2 weeks? Would it be in 5 days? And so we did this experiment where we found this area in Panama that we had heard before. We knew there were Nulls there. So we did a standard walkthrough, 90 minutes, took our standard data, species, sex, perch, body size, all that sort of stuff. What we did differently is that everything we found, we took. So we took it, threw it in a big garbage bag, make sure they were safe, and then came back. And the idea was to come back on successive days and see, well, when does this actually get back to normal? Does this extreme instance of specimen removal have a detrimental effect? Surprising to us. 24 hours, the next night we came back, you could not tell we were there. The same, actually more individuals, and then you're thinking, oh, it's a bunch of juveniles or something. No, it's adults, it's males, it's females, the same ratios. So, we've actually used this paper to kind of reply to that. Could you take 5 instead of 20 comment? And it's helped us in some cases with permitting. Okay, but let's talk now a bit about the work and a bit about anoles in some more depth. So the range of anoles is from the southeastern United States, south through the Caribbean and Central America down to South America, southern Bolivia and Brazil. So we have been to 19 countries to catch anoles. I've seen 269 of the 407 species in life. We've been to all the Greater Antilles. We've been to a couple of the Lesser Antilles. We've been all along the Andes, most of Central America. I've caught anoles in Hawaii and Taiwan. And so all these places have their own anole complements, right? We want to know about all of them. But we use these same techniques everywhere. There are huge differences in kind of how that goes. So, the Caribbean, you can collect, not see, but collect at rates of something like 1 anole per minute if you are really going for it. In some places we go, some parts of, say, the Bolivian Andes, it's more like 3 hours per anole. And so it varies a lot. And the mainland is much harder than the islands. That is a general rule, and there's been work done on that. But so how does, what do we get out of this? Well, one of the ways we do things is to go to type localities. So especially if we're dealing with a really difficult area like Pacific Mexico, in the kind of Guerrero area, has the boringest complement of anoles in the world. David Hillis once referred to anoles as a group of small brown lizards that all look the same, and I think that's because he worked in Mexico, because this is what you're dealing with. So to beat that, to sort of know what you're catching, you have to go to their type localities. So we did a type locality blitz in that area of Mexico and got 9 topotypical species in less than a week. I was looking at the notes, our most number of species in a night. I think we've done 9 in a night more than once. This is a great site in Buenaventura, Pacific Colombia, type locality of a couple different things, and just an amazing area, a lot safer than it used to be. But we were at this kind of offshore beach site, and the habitat wasn't great, but just great diversity. We found multiple new species in some evenings. The most for that is 3. This was a site in Peru. That's Enolas sweeneyi on the left, Williams Mittermeier aurum, that horribly named species on the upper right, and then Peruensis on the lower right. And I know those 2 on the right look like they're the same thing. They're actually really different in things like their male dewlap. I don't want to give the impression that we just sort of walk in and this works all the time. We say sometimes in the lab that, like, it wouldn't be as satisfying, it wouldn't be as much fun if it worked every time. So we've had plenty of failures as well. This is this species Enolas Proboscis. It was known from one specimen that Williams described in 1975. We kept going back to what we thought was the type locality. This is me. I've got the type description in my right hand, asking the herp gods why Rio Baba isn't producing for us. If you look at the type locality in the paper, 2.4 kilometers south, turns out if you look at the specimen tag, it actually says 24 kilometers south. So we were in the wrong place. We did eventually find the lizard in a totally different area, not at the type locality, but now it's been found in some good numbers by, that fantastic group at the Católica University, Omar Torres and Fernando Ayala and Santiago Ron and those guys. So we ended up okay with that one. My greatest failure as far as anole herping is this guy, Enolas roosevelti. So this is the only species of anole that may have gone extinct in human history. And so the type specimen at Harvard is shown in the upper left. The blurb from Fish and Wildlife of it being endangered is in the upper right. It was described in 1931 by Chapman Grant. He got one other specimen in 1932. There hasn't been a confirmed sighting since. There's a bunch of not a bunch. There's 8 total specimens in museums. Most of them are from the 1800s and are in European museums. It's been found on 4 islands. So from west to east here, Vieques, Culebra, St. John, and Tortola. And that's the weird thing here, right? The other weird thing is that there's still good habitat on at least 3 of those islands. So if this has gone extinct, it's gone extinct 4 places independently and 4 places that still have good habitat. There's all kinds of evidence that this thing has always been rare. Like anyone that you used to talk to who actually caught it would be like, yeah, they only, Demas only got 1 of those, and we were always collecting for granted. We only ever saw 1. So I've been to try and catch it 7 times, 7 separate trips, and I'll keep trying. We'll see. If any of y'all are in the Virgin Islands and you find it, give me a call. So we'll keep working on that one. All right, but I promised you we'd talk a little bit about kind of science and what anoles are good for. What they're famous for, of course, is evolutionary ecology, a lot of the ecomorph concept. So the idea that you can be in a room the size of the MVZ herp library and find a dozen different species, like in Saroa in Cuba, and they're all doing slightly different things so they can coexist. Along with those slightly different things, they have correlated morphologies, hence ecomorph. This concept actually started with an ichthyologist, Bruce Collette, on the left. And then Stan Rand, of course, was also instrumental, but the main architects are, of course, Ernest Williams, who really popularized it and talked about it, and then Jonathan Lassos and his extensive lineage of students have just done a tremendous job just blowing this up and adding a modern phylogenetic component to it, and just taking it in all kinds of different ways. Just to be clear, this this sort of thing does occur on the mainland as well. So this is one of our sites, El Cope in Panama, and on the screen there are 10 of the 12 anole species found within a kilometer of that field house. And so you can see the diversity of sizes and colors there. And somehow we got them to cooperate on the window so we could get a photo. A lot of our work is bringing a night component to the anole community stuff. So just like things, partition habitat and live in set places diurnally, they do it nocturnally too. Some of it reflects the diurnal behavior, some of it doesn't. There seems to be morphologies that go along with this. And before you think of it, I've already patented the word sleepomorph. So don't, that's mine. But this is just some data from a community in Panama, and you can see there's differences in their mean height and differences, and then like in their variance too. Some things are found everywhere, you can see them. Other things are just always right in a particular spot. We work on anole phylogeny, so we did our big tree stab at it a couple years ago, where we included every species, 379 at that time. There's more now. We do a bunch of comparative stuff with this. This is a paper we have coming out soon that basically shows that there's some under-evolved morphologies in anoles. Things don't evolve that have short toes and long hind limbs, for example. And you're thinking like, oh yeah, whatever, there's a developmental cascade and there's a gene that says to get longer and your limb and your toe get longer. Here's the complication. The other thing we did in this paper is that we looked at the same thing within species and the pattern doesn't hold up. So within species, having long limbs doesn't mean you're going to have long toes. There's no relationship there. So that means at least part of the story is probably selection rather than this kind of, constraint. Undoubtedly, there's some interaction there, as there always is, but neat that, maybe it's just selectively bad to have long toes and short limbs. We put together a bunch of field guide panels to help people identify these things. We have an electronic key now that's mounted. I've looked at thousands of dead anoles and collected data. And so this thing still has all kinds of problems with it, but it's useful still. This is like, okay, if you're on the eastern slope of the Andes and you find you're in Ecuador and you find a species that's pretty big body size and you punch those 3 traits in, there's only 2 possibilities. And then you can go and see, okay, I can tell them apart by their scales on the dewlap. And so we use this mostly for like really difficult preserved animals, but it should be useful kind of generally. It's on the web, as I said. Eventually, it should come out as an app of some kind. We describe a lot of new species of anoles. So these are forms from, that's Cuscoensis from Peru in the upper left, Magnifalis from Panama on the lower left, Ankykaye, Colombia, lower right. Really pretty one in the upper right is Cunayale. And then the square panel in the upper right is actually named after Miller administrator Kathy Day. So that's Enolas Kathy Day, the Miller at Berkeley. One of the really cool new species we described recently was this aquatic form. There's the aquatic anoles are just like the coolest ecomorph out there. They behave like basilisks. They will always found by a stream. They dive in the water to get away. They swim. They go underwater. They can stay underwater with an air bubble to stay around. And they sleep really weirdly for anoles. This one it's sleeping on the edge of that boulder in this splash zone. So it's loud there, it's crazy, and he's just like, okay, I think this is where I'm going to sleep. But you can see where that can have some defense for predators as well. We've rediscovered a lot of anoles, by that I mean, you have something that's only known from one specimen, or it's known, hasn't been collected in a long time. Our latest example of this is hey Sharon, our latest example of this is in the lower left there, Enolas lamari, which is one that Joseph Barnett in my lab and we went down from Bogota when we were in Colombia this last time This had been known, it's described from one specimen in 1992, and we went down and got a nice series and got some sleep data on them. One of my favorites are rediscoveries. I'm cheating a little calling this a rediscovery because it probably, it hadn't been seen in a long time, but it was already known from a few specimens. But this is Enolas Alvarez del Toro. This is the cave anole. So on the lower left is us walking inside a cave. There's these like limestone caves in the middle of pastures in kind of Isthmus of Tehuantepec area. So we went inside these caves at night with our lights and found things like these guys sleeping on the ceiling of a cave 10 meters in and like 15 meters up. You know, what the hell, man? We did go back for those during the day and found them, you know, messing around on the outside. But the really neat, you know, ecology for them there would be fun to do more with. You know, probably, you know, one of our big rediscoveries early on was this Enolas Proboscis. And so this is something we found in Ecuador. And this was a species, so this is one that it's kind of like a little bit like the occultist story. It was described in 1956 by Peters and Orcez from Mindo, or Canuco, which is right by Mindo. And so this is the place, it's like an hour and a half from Quito. People go there all the time. It's a birding spot, it's an ecotourism spot, you know, families go down there from Quito. But 1956, nobody had seen one in 50 years. And then 2005, a birding company took a photo of one on the road. You know, it obviously wasn't supposed to be on the road, but they take this, they're like, oh, that's cool, because it is. And so they take a photo and it pops up on the web. And so anole biologists are like, ah, and so we went down there to look for it. And we found a nice series and wrote a paper about it later. And then there was so we did this with our Católica folks. You know, Fernando was there helping us, and then there was another Ecuadorian group that found it about the same time, Mario Yanez Munoz. And so now, you know, this is one of those that is like, it's kind of continually rediscovered now. Like if you do a search for proboscis on the web, everybody's got a cool proboscis photo. And like, here's a news item from 2013, you know, 4 years after we found it. It's touting that somebody there found it. And this thing was like in the LA Times and became the Wikipedia write-up of it. And we're like, oh. Just an amazing anole that again, now, you know, if there's a particular road and it's not good habitat, it's just, you know, finding the right trees in these pastures there and on the edge. And it's a twig thing like a cultus. Okay, I don't want to go too long here. You know, this is a bunch of people who have helped with this. I think I still have a couple minutes. So I'm gonna end up with just talking about our herp class. We do this thing where we go on this kind of field trip with our actual herpetology class at UNM. So we take a spring break trip, and we've been to Panama a couple of times. We've been to Puerto Rico. We've been to Cuba, and we've been to Honduras. And it's just fantastic. You know, we make it, you know, we just live on peanut butter and jelly and stay in terrible places and have some nights where we don't sleep. And, you know, I used to do these trips early on. I was like, oh, we need to make this cultural, you know, let's go to a museum. Let's, you know, go during the day. And then what I realized over time was that the students, they wanted a real trip, you know, and so we end up running it just like one of our trips where they're just, you know, they're staying up all night. And it's really fun, you know, to see them respond and embrace it. And you're like, okay, we've probably got about 30 hours awake. And they're just like, oh, yeah, all right. That's what we do. And so that's been a really fun way to pass this on and experience it again. But all right, so that's all. I guess I'll take questions. I'll try and alter this so I can see people better. Should I stop screen sharing? Is that how this works?
Sure.
Okay.
Then you can see more faces. But before you start taking questions, we should all unmute and clap for you.
Oh.
Awesome.
All right. Did that just happen there? Okay.
Still applauding.
All right. Looks like, okay, there we go. Sort of. Crap. What if I, oh, hold on. There's a stop share button. Do you think that would work?
I could force you to stop.
Yeah.
Yeah.
Do you do much teaching, Steve?
Not with
Need
Zoom.
to do more learning, apparently.
Well, cool. Do we have questions for Steve?
I definitely thank you so much, Steve. That was amazing. I was just wondering, finding them at night, have you come up with ways to then use that data to learn something about their diurnal ecology, or is that still kind of a mystery? Like, where are these things hiding during the day?
No, it is, and that's actually what you mentioned is it's kind of a standard technique has become for studying a lot of the rare things. Like you'll go and something like the Enolas Anoles in Cuba are just another one of these. They're you just don't see them during the day, and they're spectacular. And so there's a lot of kind of odd ecology behavioral work on these things where you go and you find one, and you tie a ribbon next to it or whatever, and then you go to bed or do whatever you're doing. Then you show up before the sun comes up, and then you just track it and watch what it does. And so people have been able to do that, to kind of parlay this work with rare things to figure out what they're actually doing during the day.
Super cool. Awesome.
So I noticed you have a very good set of trait data for many species. This is an ongoing conversation we've been having at AmphibiWeb that much predates me. Are you connected at all with people building trait databases? Because I feel like your data set is gold.
I'm not really. It's more I've given it out to a lot of people, and it is really like we worked a lot on it, and we have data for a set of traits for every single species of anole, really. And it's just for the basic set, it's it's like hind limb length, head length, body length, a scale trait, scale size trait, basically. Is that it? Head, tail, or head, and then and then if you get farther out, then we have not quite for everybody, but things like tail length and toe length and things like that. But no, I'm not hooked into it's not on any kind of big public database or anything like that.
Yeah, I'd be interested. There is an NSF effort right now called Futres. I'll pop a link into the chat if you're curious. I'm not sure if they're actively looking for data sets, but it sounds like maybe it's something they might be interested in.
Yeah, no, that'd be great. I didn't know about that.
So Steve, how often do you see anoles that are too high for you to catch with your pole?
It does happen. It does happen, and we actually the thing that we fall back on for that is, and I used to have a student in my lab who had a budding pro baseball career, and so what we do in that situation is we'll attach a long length of really long length of fishing line to something relatively heavy, and try and throw it up over the branch. And so we can reach up to this being a 20-foot pole, we can reach up to 7 or 8 meters or something like that. But when something is a little bit higher than that, but there's a branch you can see, we'll try and loop some line over that and then shake the branch that way and see if we can get the thing to fall. That's really what we fall back on. It doesn't work that well, but it's when you're frustrated, it's worth a try. Maybe it's 50%.
50% is pretty good.
Yeah.
I was going to suggest the super blowgun.
I know. Well, that's I almost had a section on that with that. I remember Jimmy used to sit in the hallway at the Castetter, and he would had this blowgun that he would use to shoot down the hall for target practice. And one time he got Eric Pianka in the eye, and oh my God, Pianka just lost it.
He did not.
He's classic for these sorts of stories. That's right. I killed a famous herpetologist with my blowgun.
Follow-up question there, Steve. So I'm reading this book over by this guy named Lee Grismer of talking about all this agamic lizard behavior on the peninsula in Malaysia. Because a bunch of these facts that he worked on turned out to be really closely related to things in the islands nearby. And you read the natural history sections in a book like this, and it's full of statements like these may be, these are active diurnally. You see them out during the day on the backside of trees away from the sun. And these are agonocephalous, right? These sort of big active agamids. And I haven't ever seen the natural history that he describes of those animals on the peninsula are completely in opposition to, all of my 30 years of experience with their closest relatives on the archipelagos, which is that we find them at night sleeping and we never see them during the day. Like they see you coming long before you get there, you don't see a thing. And so I'm wondering, does Lee Grismer have much better eyes than I do? Or probably, or do people like you and me just never go out at 8 a.m. in the morning and really ground test, ground truth the other side of the coin? And I guess I'm wondering,
No, I have that. I've had that same thought, 100 times, Rafe, and we have sometimes tried to ground truth this, but it gets like, pretty rapidly, it gets frustrating. Like I've had students that I sort of commissioned to do that, be like, okay, do the day, do the night. And they talk to me a week later and they're like, I can't freaking see anything during the day. What are you talking about? The other thing, so we try and run that experiment once in a while just to fact check ourselves. The other thing I'll say is that there are, there's a handful of species that at least in anoles that genuinely are easier during the day. And so things that tend to be ground anoles. So something like Tropodonotus or Humulus, these are things that are often marked to be the most abundant thing in an area. So be like, oh, there's Humulus, there's one every square meter or something like that. We never see Humulus because they sleep mostly in the leaf litter. And so, they're doing something fundamentally different at night that makes them rare to us. And the stuff that is common to us, is very rare to the people during the day. So I think there's some of that too, where there's, it's not just a kind of trade-off in skill set. It really does have something to do with the biology of different species.
Not just search image, yeah.
I just want to add that, flying lizards are like the reverse of Enolas, because I do a lot of night collecting, but I also do a lot of day collecting. And if I see one Draco at night on a trip, I would say that's the max. I mean, we just don't see them at night. And often when we do find them, they're on tree trunks, like they sleep on tree trunks, unlike the Anoles.
Yeah.
I think they're way up there. They're not down low.
Yeah. There's
Can I say, when George Gorman and I were working on the Enolas of the Caribbean, and we collected on virtually every Caribbean island, caught every known species, we only worked during the day. The only thing we ever collected at night was pulculus on Puerto Rico, where you collect them on the grass stems. But everything else was during the day. We got every species that we were looking for. So they're there during the day, I can assure you.
The other thing, well, it's probable your eyesight is better than mine, but I will also add that the Caribbean is fundamentally different than the mainland.
That's perfectly possible, yeah. I mean, it could have been we would have caught more of them at night. We'd never even occurred to us to go out at night. Because once you sort of know what kind of habitat they're in and whether they're on the trunks and that sort of thing, it made it easier to find them, knowing where they're going to be. I don't know.
Sure.
Can I ask a question, since Rafe talked about Southeast Asia? I feel like I can now transition a little more without feeling awkward. Great speech, by the way. Great talk, Steve. I really enjoyed it.
Thank you.
So my question is, what's going on with the Pinocchio morph? Is there like a, because we have that independently evolving even in Indonesia, like Southeast Asia. We have this, Harposaurus modigliani. There's another species that we are currently describing. I mean, by we, I mean, the Eric Smith group at UTA that has the same, Proboscis. So is there like an evolutionary reason for that, or is there an hypothesis?
Yeah, so there's been, the group in Católica in Quito has done some things with that, where they've put males and females together and see how they use the proboscis. So like these things, they still have a dewlap in both males and females actually, and it's not very big and it's drably colored, but they end up using both the dewlap and the sort of standard lizard push-ups, and they do like a wave with the proboscis. So beyond it being kind of part of the display. I don't know if I don't think they've done the work to show that it's something like, increasing the virtual size to win a contest or something like that. I don't think they've gotten that far with it, but it's certainly used in their mating displays.
So it's just the mating display.
Yeah, that's all. It's never, for what they've observed, they've only seen it used by the male to, no. I know it's been used by the male to attract the female. I don't think it is, you see, now I'm not sure. I'm not sure. I know it's not used as like a weapon because it's too soft,
Right.
but it may be brandished in particular ways when it's male as well. I'd have to check on that.
Right. That's cool. So it'll be nice to have comparative data maybe from Southeast Asia at some point.
Oh yeah.
It's also a male trait in Southeast Asia with Aphaniotis and Thematorhynchus and Harposaurus, these things that converged on that, the agamids that converged on it. They're also catatonic, right? Like slow walkers.
Yes. Yeah.
And so it seems like that it goes with that.
Yeah. It's I remember that in the original, I don't think it was in the proboscis description, but in Williams' review of the Proboscis Anoles, he had figures of a couple of those agamids that had the same process. But that's interesting if they're all the kind of slow walking behavior.
Thank you.
Yeah. Hey, Steve. Good to
Yeah.
see you again.
Hey, Catfish.
Yeah, so I just sort of adding, anecdotal comments. When I was a postdoc at Berkeley and I was down at Tapanti in Costa Rica, Jonathan Lossos came through after an OTS course, I think, and he was just starting to, almost like starting to think about working on anoles, or maybe it just started. And I took him out at night, and just like walking along the road, we got 7 species one night.
Nice.
And when he was there, he saw almost nothing during the day, except maybe like leaf litter, whatever the little brown thing in the leaf litter is there.
Humulus guy.
Yeah. But yes, I mean, that's totally it. On the mainland, you really, you just see so many more species at night, I think, than during the day.
Yeah. I'd like to think that's always your computer background there, Catfish. So I'm gonna that thought.
So you're actually holding a coach whip here, Steve.
Oh.
Circa 1995, I think, for those of you who you don't look much different, man. You still got it. You still got it.
Yeah, I think I was 10 years old then. I think that makes
We have more questions for Steve. I had like 5 things that I noted while you were talking that I wanted to ask about, and now I've forgotten them.
Well, I really appreciate everybody coming by. Like I said, it's a thrill for me to be here, even virtually, and get to see y'all. Man, it's really cool. I'm gonna have to get out there in person at some point. So fully vaccinated now, so got that going for me.
Steve, I thought of one more question. So I've been mostly studying these slender salamanders, Batrachoceps in California, and we do have a few spots where you go back time and again and you think they're there and you can't quite find them. What advice do you have for anybody working on something that is just a pain to find? And either how do you get other people interested to fund you to look for them, or how do you just keep yourself going when it's a situation like that?
I've been on one of those with Dave. I can't remember what Batrachoceps it was, but we're doing a big loop and looking for Batrachoceps, and we went there and it wasn't there, and you could tell Dave didn't think we were going to find it, but but it had been found there, so that was the key. So we had to go back. So I know what you're talking about, and I find those points fascinating, actually, that to be on that loop that it's been there either because you found it or there's a specimen from there that you trust, and it's like, okay, a week later I'm not seeing it. I'm supposed to be at least competent at this, and I can't, I'm just not. I know it's here and I'm just not seeing it. So I think I've got some one of the, I think in that collecting chapter I've got some treatment of this, right? You know, the conclusion is just that sometimes you just have to put in the time, unfortunately.
Roosevelt.
That sounds like where I've come to about it. So
Yeah, got plenty like that.
I've made multiple attempts for campi. I've never found it.
Right.
Patrichoseps campi.
oh, that's not even a hard one, Jim.
But I wasn't there for that long.
Well, Jim, we did drag you to Nevada de Colima, which has been 115 years since anyone found it. So
Yeah, I got to participate in that field effort.
Anytime I'm on a group like this, like if you talk about, like you give a talk like I just talked, or you say what like Jim did, you're setting yourself up to like somebody's going to be like, you couldn't find parlous, really? You know, like God, we were out there about a half hour and we got one. You know?
That's because you had the Ace Beam K30 GT SBT 550 max lumens light. Thanks for the tip. I'm gonna buy one tonight.
Got to compare notes on our headlamps.
No, I mean, I'm always interested in hearing about, you know, what kind of lights and stuff other people use, because it's evolved. Like we just started using that KT just really recently because we had just fallen for the night mountain biking lights, and it finally got to the point that some of these other lights actually do have a lot more power. So for certain situations, you know, if something's really high or you just got to have one of those in your quiver at least.
I think the brightest headlamp I've ever used was the Night Rider, which is another one of those bike lamps.
Yep. Yeah, we've used Night Rider before. We've kind of settled on this company Dynote because they have just excellent customer service. You know, they know us, like, oh, you're the lizard guys.
Yeah, the Night Rider has this nasty habit of shutting off right when I'm jumping off a log or something.
Yeah.
Little frustrating.
Yeah.
Fair warning, though, referred to it as the Eye of Sauron.
Yeah, there's no, I mean, the technology just goes incredibly fast with this stuff. I mean, it's just, you know, the next time you get a 10,000 lumen light, there's going to be a 20,000 one next week. And it's just ridiculous. And I think we are, at least, I mean, I feel like we're at a point that I don't want my light to be any brighter, and I seldom use it on its brightest setting because, you know, you're looking for the contrast and you don't want to wash everything out and have it look like daytime.
So Steve, what fraction of your students that take your course actually go on these field trips, and how are they paid for? Do they pay their own way?
They do, yeah. You know, we've tried to point them towards, there's some small grants at UNM, but, you know, we mostly just try and make the trip as affordable as we can. You know, in the past when I've had grant money, I've thrown some in there, or when we're going somewhere that's a little pricier, I'll get my chair to throw some money in because, you know, we don't have a rich student base at UNM. So we mostly just, like, if we can, you know, we've done trips where we go to Panama for like 9 days and charge them $1,100, you know, including the plane ticket. And so it ends up being about, you know, if we have 14 people in the class, you know, maybe 9 of them go on the trip.
Wow.
And it's really, because some people just take the class for the trip, and others, you know, really like the material, and some it's kind of a combination. So it varies, you know, and depends on where we're going too. You know, everybody wanted to go to Cuba.
I'd like to enroll in your class.
We'd love to have you. You'd be like a ringer in there, you know, like, who the hell is this guy?
This guy in the back of the room. Yeah.
What's up with that blowpipe? Awesome.
Well, I guess maybe at this point we can let you off the hook, Steve. But unless anybody else has any further questions, but I'll stick around for a few minutes if you care to hang around, Steve. I have things I'd like to chat with you about.
Yeah, no,